the Medical College of Wisconsin, the Max McGee Research Center for Juvenile Diabetes. His talk today is monocyte populations in type 1 diabetes families exhibit high activity and subset abundances that correlate with clinical progression. A little bit about him, his bio. He's working as a postdoctoral research fellow in the Department of Pediatrics, Division of Endocrinology and Surgery at Medical College of Wisconsin, as I mentioned. And he received his doctorate in 2019 from Anna University, Chennai, India, where he focused on investigating long non-coding RNAs, LNC RNAs, linked RNAs, mediated mechanisms, particularly related to cardiotoxicity under hyperglycemia conditions such as DCM, diabetic cardiomyopathy. And after graduation, he joined Dr. Bosnack's lab as a postdoctoral research fellow in the Department of Medicine at the Medical College of Wisconsin and continues studying these linked RNA-mediated mechanisms of impaired cardioprotection under diabetic conditions. Then in 2021, he joined Dr. Martin Hessner's lab as a postdoctoral fellow at the Department of Pediatrics Endocrinology, still at Wisconsin. In his current position, he's really looking into the genetic and immunological basis of familial inflammation associated with type 1 diabetes susceptibility and how levels of innate inflammation impact the rate of disease progression. Further, his research involves the usage of probiotics to promote immunoregulation and possibly slow T1D progression before and at T1D onset. Dr. Pant has published more than 15 manuscripts in well-reputed international journals, Science Advances, Redox Biology, Antioxidants, Anesthesia and Analgesia, and scientific reports, and he has 10 plus years of expertise in diabetes research. He's also teaching, mentoring students, and we're very excited to have him here today to discuss this work. I would ask people to drop questions during the talk into the chat. We'll take those questions during the talk, and also he'll field some questions after. Thanks so much for joining us, Tarun. We're looking forward to this.
Well, thanks a lot, Dr. Wesley, for that introduction. And I'm more than happy to present my work at the Sugar Science Podium. So with this, I'm going to begin my presentation. A very good morning and good afternoon to all the listeners. In this presentation of mine, I would be talking about my work that describes the role of monocyte population in type 1 diabetes. Well, as most of us here are familiar with the fact that type 1 diabetes is a heterogeneous autoimmune disease that arises from immune cell-mediated destruction of pancreatic beta cells. Well, here is an image on the right that shows the infiltration of the leukocytes, specifically CD3 positive, CD8 positive cytotoxic T cells into the pancreatic islet, where they mount an immune response against the beta cells, eventually leading to their destruction. Now, this immune cell-mediated destruction of pancreatic beta cells results in insulin deficiency and a lifelong dependency on exogenous insulin replacement therapy. Now, there have been numerous prior studies that have shown that type 1 diabetes pathogenesis involves adaptive immune responses. For instance, there have been immunotherapies used targeting T cells to slow down type 1 diabetes progression prior to clinical onset. However, a growing body of evidence support a role for altered innate immune response in type 1 diabetes pathogenesis. Now, there have been studies suggesting that genetic and epigenetic, along with the environmental factors, can activate innate immune system, further aggravating type 1 diabetes pathogenesis. Also, there have been studies suggesting expansion of specific immune cell subsets like monocytes, which can trigger inflammation, subsequently reducing the beta cell function to promote type 1 diabetes pathogenesis. Now, monocytes are innate immune cells that comprise 10% of the nucleated blood cell count and are classified into 3 phenotypically and functionally heterogeneous subgroups. This classification of monocytes into these different subgroups is based on the expression of surface marker proteins like CD14 and CD16. Now, to begin with, they can be classified into 2 major subgroups based on the expression of CD16 marker, which include CD16 negative classical monocytes and CD16 positive intermediate and non-classical monocytes. Well, the intermediate and non-classical monocytes can further be distinguished from each other based on the expression of surface marker protein CD14, where intermediate monocytes are CD14 high and non-classical monocytes are CD14 low. Talking about the functionality of these monocytes, the classical and intermediate monocytes are primarily involved in antigen presentation, T cell stimulation, and mounting inflammatory responses. whereas the non-classical monocytes exhibit both inflammatory as well as anti-inflammatory behavior. Now, in addition to these traditional subsets of monocytes, more recently, single-cell RNA-seq has fostered the identification of new monocyte population. Now, using this approach, a research group, Walani et al., defined 4 subsets of monocyte in healthy human subjects, where the intermediate monocytes were broken down into 2 new clusters designated as mono 3 and mono 4, whereas the mono 1 and mono 2 cluster contained traditional classical and non-classical monocytes. In addition to this, another research group, Yu et al., studied monocyte heterogeneity in autoimmune uveitis using single-cell RNA-seq, where they identified 6 new subpopulations of monocytes. To specifically point out, they're one of the population designated as NK-like monocyte were very similar to mono 4 in the terms of their transcript expression. So both mono 4 and NK-like subsets expressed a cytolytic transcriptome, which include high expression of transcripts related to cytolytic proteins such as grain enzyme, perforin, and granulolysin. Now, while an understanding of the monocyte heterogeneity in human health and disease has significantly increased, the studies examining the abundance and functionality of monocytes in type 1 diabetic patients and their at-risk relatives remain limited. And hence, that's what our study focuses on. Our study includes cross-sectional cohort having groups such as unrelated healthy controls. The subjects belonging to this group have no family history of type 1 diabetes. In our study, we also have autoantibody negative healthy siblings of type 1D patients, which are designated as high-risk SIPs and low-risk SIPs. The subjects belonging to the high-risk SIP category have a higher susceptibility to develop type 1 diabetes as they possess DR3, DR4 haplotype, whereas the subjects belonging to the low-risk category have a lower susceptibility to develop type 1 diabetes as they are non-DR3, non-DR4. In our study, we also have type 1 diabetic patients. The subjects belonging to this category have been recently diagnosed for type 1 diabetes. However, they have achieved a better metabolic stabilization in this time window. And then finally, we also have long-standing type 1D, which includes subjects with type 1 diabetes for more than 5 years and have little or no endogenous beta cell function left. Now, there have been studies in the past that have suggested that unfractionated PBMCs of type 1D family member hypersecrete pro-inflammatory cytokines such as IL-1 alpha and TNF alpha post-LPS stimulation. To further characterize the innate immune activity in the type 1D family member, we purified the monocytes from the cryopreserved PBMCs across a cross-sectional cohort. These purified monocytes were further being stimulated with lipopolysaccharide for 24 hours, and later we quantified the secretion of pro-inflammatory cytokines, including IL-1 beta, IL-6, and TNF alpha in the conditioned media using ELISA. Our results showed that there were no difference in the secretion of these 3 pro-inflammatory cytokines among the type 1D family member post-LPS stimulation from their monocyte, which suggests that the HLA-DR haplotype has no response or has no role in the production of these pro-inflammatory cytokines from these LPS-stimulated monocytes of the type 1D family. However, when we compare the results of type 1D family members to unrelated healthy control, we observe a significant increase in the secretion of these 3 pro-inflammatory cytokines from the LPS-stimulated monocytes of type 1D family member compared to the monocytes of unrelated healthy control. Now, this observation here suggests that the monocytes of type 1D family member are hyper-responsive to LPS stimulation or TLR4 ligation compared to the monocytes of unrelated healthy controls. Later, we also looked into the transcriptomic signature of these purified monocytes in a cross-sectional cohort using FE matrix array. To begin with, we identified 826 probe sets, which were differentially regulated across a cross-sectional cohort. Further, the hierarchical clustering of this data set suggested that the transcriptomic signature of the CYPs had an intermediate relatedness to that of unrelated healthy controls and type 1 diabetic patients, as shown here in the heat map. Further, this analysis also suggested that the transcriptomic signature of the monocytes was highly distinct from that of type 1 diabetic patients, as shown here. Further analysis of this data set also indicated that the monocytes of type 1 diabetic patients had enrichment of certain transcripts which were found to be associated with natural killer cell activation and natural killer cell cytotoxicity. Now, interestingly, this monocyte-specific transcriptomic signature observed in the monocytes of type 1 diabetic patients had a high identity with the transcriptomic signature also observed in the mono4 and NK-like clusters in the past, which suggests that the cytolytic transcriptomic signature of the monocytes of the recent onset type 1D had overlap with that of mono4 NK-like subsets. Now, considering that single-cell RNA-seq has fostered the identification of novel monocyte populations in health and disease, we use this approach to look into the immune cell dynamics using the cryopreserved PBMCs across a cross-sectional cohort. To begin with, we identified 13 distinct clusters based on the transcript expression. And to specifically point out, we identified cluster 0 and cluster 7 to be CD14 positive and CD16 positive monocytes. Now, later on, these two clusters were reanalyzed, and similar to the studies in the past, we observed high heterogeneity as evidenced by the identification of 10 new clusters among total monocytes. To specifically point out, all of these clusters expressed CD14, CD16, as well as HLA-DR marker as shown here in the right in the U map. However, to specifically point out, among these 10 clusters, it was cluster 9 that had the maximum transcript expression which shared identity with previously defined mono4 or NK-like subset population. Now, some of the transcripts which were being expressed in these cluster 9 monocytes and also in mono4 or NK-like subsets reported in the past are shown here in the U map, which includes markers like KLR-B1, KLR-D1, and perforin-1. Now, next we were interested in quantifying the average expression level of these transcripts at single cell level. And to do so, we calculated something that we refer to as the gene module score or mono4 enrichment score. So to do so, we began by identifying the transcripts which were dysregulated in our group, in our study, as well as that also overlapped with Vallani's mono4 cluster and use NK cluster. Our results here showed that among these 10 clusters, it was cluster 7 and cluster 9 that had the maximum enrichment score for these mono4 transcripts. Also, interestingly, our results showed that the type 1D patients compared to the autoantibody negative CYPs and unrelated healthy controls had a significantly higher mono4 enrichment score in most of these clusters, which include cluster 9 and cluster 0 through cluster 6. Now, this observation here suggests that the monocytes of type 1 diabetic patients in general exhibits the cytolytic transcriptome, which shares a high identity with the natural killer cells transcriptomic signature. Later, we also specifically looked into the cytolytic transcripts among these 10 clusters, and we also were interested in investigating that how the expression of these cytolytic transcripts change across our cross-sectional cohort. Our result here showed that the type 1D family member had a higher expression of these cytolytic transcripts compared to the unrelated healthy control across all these 10 clusters. And this expression of these cytolytic transcripts was found to be most abundant in the type 1D patients. And this trend was found across all the 10 clusters. Now, this observation here suggests that the type 1D family members' monocyte exhibit a cytolytic transcriptomic signature. In addition to looking into the cytolytic transcripts, we also looked into the chemokine cytokine transcripts in all these 10 clusters. And also, again, we looked into that how this expression pattern changes across a cross-sectional cohort. Now, our results here showed that in a cross-sectional cohort, the type 1D family members had the highest expression of these chemokine cytokine transcripts compared to the unrelated healthy control. However, we had an interesting observation here that in a cross-sectional cohort, it was the CYPs which had the least expression of these chemokine cytokine transcripts, which suggest a state of immunoregulation or indicates that these autoantibody negative CYPs are better immunoregulated. Well, later, we also validated our preliminary findings from our single-cell RNA-seq and Effie-matrix array experiment using flow cytometry. So using flow cytometry, we were interested in looking into the abundance of the traditional monocyte subsets, which include classical, intermediate, and non-classical monocytes in a cross-sectional cohort, as well as the newly defined mono4 or NK-like subsets. So I'll briefly talk about my flow cytometry panel here and my gating strategy. So to begin with, I'm gating over the PBMCs here, and then to exclude the non-viable cells in our antibody panel, we have viability dyes. So I'm gating over the viable cells here. And then in my panel, I have CD3 antibody for the CD3 marker expressed over T cells. And then I have CD19 for B cells. So I'm gating over CD3 negative, CD19 negative cell population to exclude the T cells and B cells from the final population that we are interested in. Later, to exclude the natural killer cells from our monocytes, we included CD56 antibody, and I'm getting over CD56 negative cell population here. And considering that the monocytes play a crucial role in antigen presentation, so they have a high expression of class 2 MSC or HLA-DR, so we included an antibody against the HLA-DR, and we are getting over HLA-DR high cell population here to finally distinguish or segregate the traditional monocyte subsets based upon the expression of CD14 and CD16, which include classical monocytes, intermediate, and non-classical monocytes. Our flow cytometry results showed that the type 1D family member had a higher circulating abundance of classical, intermediate, while a lower abundance of non-classical monocytes when compared to the unrelated healthy control. This difference was found to be significant when we compare the recent onset type 1D patients to unrelated healthy controls for all these three subsets. In addition to this, we also looked into the abundance of these three subsets in the long-standing type 1D patients, and we observed a significant decrease in the classical as well as a significant increase in the non-classical monocytes in the long-standing type 1D patients when we compared them to recent onset type 1D. Now, both the long-standing type 1D patients as well as the recent onset type 1D patients, they exhibit a similar glucose level. So by which I mean that this increased abundance of the classical, inflammatory classical and inflammatory intermediate monocytes which we observe in our recent onset type 1D patients, is not a result of elevated glucose. In addition to this, we also looked into the circulating abundance of natural killer cells in our cross-sectional cohort, and our results here showed that there was no significant difference in the circulating abundance of the natural killer cells. Interestingly, we also observed an inverse correlationship between the circulating abundance of intermediate monocytes to the rate of C-peptide decline. By which I mean that those recently diagnosed type 1D patients which had a higher abundance of intermediate monocytes at the time of diagnosis or at baseline, they experienced a more rapid rate of C-peptide decline during the 24-month study period. Now, negative is the slope here, the faster is the rate of C-peptide decline. We also observed a direct correlation between the circulating abundance of non-classical monocytes to the rate of C-peptide decline. So the individuals which had a higher non-classical monocytes at the time of diagnosis, they had a slower rate of C-peptide decline. And this relationship was even more robust when considered as a ratio. So those patients which had a higher ratio of intermediate to non-classical monocytes at onset, they experienced a more rapid rate of C-peptide decline. So our results here suggest that intermediate monocyte promote more rapid post-onset progression. Non-classical monocytes may be functioning in an immunoregulatory capacity to slow the post-onset progression. And those individuals which have a higher intermediate to non-classical monocyte ratio at onset, they will experience an accelerated decline in the beta cell function. In addition to looking into the traditional subsets of monocyte, we also expanded our flow cytometry panel to identify the newly defined mono4 or NK-like monocyte population. So to do so, we included surface markers such as KLR-B1, KLR-D1, as well as perforin-1 to our already existing flow cytometry panel that we used prior to look into the abundance of traditional monocyte subsets. So the idea here was to identify the percentage or distribution of these mono4 or NK-like monocytes in the classical, intermediate, as well as non-classical monocytes, and then also to look into the percentage of these monocytes among the total monocyte population, which we do by merging these three gates. The way we are defining our mono4 or NK-like monocyte in these three different subsets and among the total monocyte is by the combination of surface marker KLR-B1, KLR-D1, and by the combination of surface marker KLR-D1 and perforin-1. In this specific slide, I will be just representing the data of the percentage of mono4 or NK-like monocyte among the total monocytes. And again, the way we define our mono4 monocyte here is KLR-D1, KLR-B1 high, and KLR-D1 perf1 high. So our flow cytometry results suggested that compared to the unrelated healthy controls, the recent onset type 1D patients had a significantly higher abundance of these inflammatory KLR-D1, KLR-B1 high, as well as KLR-D1 perf1 high mono4 monocytes. Also, we investigated the abundance of these mono4s in the long-standing type 1D, and we observed a significant decrease in the mono4 monocytes among long-standing type 1D patients. Well, next, we were also interested in looking into the abundance of these traditional monocyte subsets, as well as the newly defined mono4 or NK-like population, in addition to the Tregs among the non-progressors and progressors, by which I mean that we were interested in understanding this immune cell subsets in the preclinical onset time duration. So to do so, we had non-progressors, which include autoantibody negative healthy siblings of type 1 diabetic patients, but they don't progress to develop type 1 diabetes. We also include progressors and the subjects belonging to this category transition from stage 0 to stage 1 or stage 2 or stage 3. So the way we describe these stages is based on the circulating levels of islet autoantibodies and how well their glucose levels are regulated. So a subject in stage 0 are autoantibody negative with normal glucose level. Subjects in stage 1 have more than 2 islet autoantibody, and again, they are well regulated as they have normal glucose level. The subjects in stage 2 have more than 2 islet autoantibodies, but they are dysglycemic, and stage 3 subjects are at clinical onset, which reflects hyperglycemia. So our longitudinal analysis included 22 high-risk non-progressors, which were examined at 2 time points, and the mean duration between the sampling time point 1 and sampling time point 2 was 5.4 years. Our progressors cohort included 17 progressors, which were again examined at 2 time points, and the mean duration was 4.2 years. Our flow cytometry results showed that the non-progressors had a temporal decrease in the circulating abundance of classical as well as intermediate monocytes over time, which was significant. We did not observe any significant changes in the circulating abundance of non-classical monocytes in the non-progressors. However, when we considered taking the ratio of intermediate to non-classical, we observed a temporal decrease in the ratio over time. Talking about the progressors, we did not observe any significant changes in the circulating abundance of these traditional monocyte subsets over time, as well as we also did not observe any significant changes when we took the ratio of intermediate to non-classical among progressors with time. In addition to looking into the traditional subsets of monocytes, we also looked into the circulating abundance of T regulatory cells. And the way we are defining our T regulatory cells here is based upon the expression of surface marker CD45RA and intracellular transcription factor FOXP3. So the way we define our activated Tregs are CD45RA low and FOXP3 high. So among the non-progressors, we observed a temporal increase in the circulating abundance of regulatory T cells. We were also interested in investigating the relationship between the inflammatory monophore and regulatory Tregs, which we did by taking the ratio of KLR-D1, KLR-B1 high monophore and KLR-D1 high, perf1 high monophore to activated Tregs. And among the non-progressors, we observed a temporal decrease over time for both the KLR-D1 high, KLR-B1 high, and activated Treg ratio, as well as KLR-D1 high, perf1 high activated Treg ratio. For the progressors, we did not observe any significant changes in the circulating abundance of activated Treg over time. And also, we did not observe any significant changes in the ratio of monophore to activated Tregs. So with this in conclusion, we can say that our study indicates that monocytes of type 1D family member hypersecrete pro-inflammatory cytokines upon LPS stimulation. And among recent onset type 1D patients, most of the monocyte subset expressed a pro-inflammatory and cytolytic transcriptomic signature, which overlapped with the previously defined monophore or NK-like subpopulation. Compared to the unrelated healthy controls, our flow cytometry results confirmed the elevated abundance of inflammatory classical, intermediate, as well as mono-4 monocyte among recent onset type 1D patients. The intermediate to monocyte ratio among type 1D patients at onset significantly correlated with the rate of decline in beta cell function. And the non-progression was associated with temporal decrease in the mono-4 or NK-like monocyte and increased abundance in activated Tregs in the periphery, as well as decrease in the intermediate to non-classical monocyte ratio. The future questions that we are interested in is, can reduction in this mono-4 or NK-like pro-inflammatory activity can slow the progression or reduce the type 1D susceptibility in those individuals which are at risk? Well, with this, I would like to specifically thank my mentors, Dr. Hessner, Dr. Chen, and to give me this opportunity to present this work and do this study. And then I would also like to thank my lab members who helped me out in this study, as well as the funding resources. And with this, I'll be more than happy to take any questions.
Thank you, Tarun. That was a beautiful presentation. We have a question from Peter Gottlieb. How many studies were in the ROT1D cohort? And has that been verified in another cohort?
How many subjects we had?
Mm-hmm.
So if I can recollect, in total, we had around close to 25 subjects.
That's a
that we
recent
had.
onset, just a recent onset.
Yes.
Mm-hmm. Okay, great. And then has that been verified in another cohort, he asks?
In addition to that, yes, we also had some additional subjects that we have been doing. So yes, the answer to that question is that in addition to those 22 subjects, we also have had other recent onset type 1D patients in which we have been investigating the abundance of these monocytes as well as other immune cells.
This is kind of a, not really a technical question, but I mean, how did you recruit for this study?
Well, so the recruitment for this study has stemmed from a long program that Dr. Hessner's lab has been working on. And in this study, actually, we have been so far recruited more than 600 type 1D family members. And this program has been going on for the last 2 decades. And in my belief, I think that we are among few groups which have been able to successfully recruit so many type 1D families.
I agree, and that's why I wanted to highlight it. So I think that's really great. Here's one from Kiati Gurda in Boston. Great talk. The chemokine increase in monocytes in RT1D is interesting. Did you compare these different autoantibody positive patients? And if so, does that have any correlation with age?
We have, in my study, I haven't looked into the correlation between the circulating abundance of these pro-inflammatory cytokines to age. However, at this moment, we in our lab are also doing certain studies where we have these type 1D patients where we are looking into the correlation of these cytokines with age. So yes, I mean, we are doing that at this moment.
Great. And then here's another question. Peter Gottlieb, agree. Great talk. Very interesting. How does your observation of monocyte changes compared to Marty Hessner's previous observation with more variable levels of inflammation in these populations? And you can just read that yourself too. It's in the chat, Tarun. You need to process it.
Does your observation of monocyte changes compared to Marty's? So the way I understand this is when more variable levels of inflammation, by which I think that what Peter means here is the heterogeneity that we have been observing. Is that I'm not exactly able to follow this, but the way I'm understanding this is that even among the CYPs as well as among the type 1D patients, we do observe a heterogeneity. And by which I mean that there are certain patients which are more inflamed compared to the other. So they can have a different level of these pro-inflammatory monophore monocytes as well as the traditional monocyte subsets, by which I mean that those patients which are more inflamed compared to the one which are less would be having a higher abundance of these monophore. That's what we believe in.
Peter, you're welcome to unmute yourself and qualify your question if you'd like.
Yeah, thanks. I think it'll make it easier. Great talk, really interesting. So, Marty's obviously, and he's on, I assume, has put on, put out the inflammatory signature, which, has been very interesting and
Yes.
obviously something that we track. And has, again, sort of, divided the populations into different groups a little bit based on that. How does your observation, how does this mono-4 thing fall out with that? Is it, does it just work in terms of, the higher number fits the higher inflammatory, or is there actually differentiation between those who exhibit this and one of Marty's populations versus the other one?
So yes, Dr. Hessner's lab in the past have looked into something that is called the composite II, if that's what you're referring to. And we haven't looked into the correlationship between the circulating abundance of monophore to the composite II at this moment, but we would be doing that. But again, what we would hypothesize there would be that those patients which would have a higher II, they should also have a higher abundance as well as functionality of these monophore monocytes as well as the traditional subsets.
Right. Yeah, that's what I was driving at, because that would obviously be very interesting.
Thank you. Here's another question, again from Boston. I'm wondering about your linear regression data of non-classical to classical C-peptide. 3 subjects stand out. Did you normalize other factors in that data, such as sex, age, or HLA type?
To my belief, I think that yes, we did.
Okay, great. So maybe you guys can communicate offline and share that.
And then again, just to mention that there is a possibility, because one of our ongoing study, which is done by one of my friend and colleague in Dr. Hessner's lab, is to look into the endotypes among the type 1D patients. And based on our study so far, we have clearly identified 2 subgroups in which one of the subgroup is considered to be more inflamed compared to the other one. So there is a high possibility that there might be subjects which sit above in that ratio of intermediate to non-classical. They might be belonging to that subgroup which is more inflamed, which we refer to as endo 1. So the major difference between those 2 endotypes is that one of the endotype is more inflamed than other. So in that rate of C-peptide decline curve, there is a possibility that those subjects which are having a more rapid rate of C-peptide decline and also having a higher abundance of intermediate to non-classical, they might be belonging to that group.
That's very good. Another question, increase in KLR-D high and KLR-RD high population. Do you have a plan to study that in NOD mice if these cells have any role in T1D?
Yeah, so that's a very interesting question. Yes, we are interested in studying the abundance as well as the functionality of these monophore monocytes in the NOD mice. So yeah, and how that changes, but it would be also more interesting to see that if in some way we can modulate the abundance of these monophore monocyte and then set up an incidence study to see that if it's having any changes in the type 1D incidence. Although we are also trying to do that with the probiotics because we have been having this ongoing clinical trial where we have administered Lactobacillus plantarum probiotic to the recent onset type 1D patients. And at this moment, we are interested in checking that if the administration of this probiotic can have an effect over the KLR-D1, KLR-B1, or KLR-D1-perf1 monophore abundance as well as functionality.
Wait, so you're postulating that perhaps this Lactobacillus plantarum could affect these NK-like cells?
Yes, so that is one of the things that we are postulating, and the reason for that is in the past we have had a trial where we administered a multi-strain probiotic referred to as BSL-3 to the autoantibody negative CYPs, and that study showed that the probiotic administration of those to those autoantibody negative CYPs declined the systemic inflammation, which again we quantified by looking into the circulating levels of cytokines, and they were decreased. But interestingly, we observed there that there was an increased abundance of single chain fatty acids, and to specifically point out butyrate and propionate. And there have been studies in the past which have suggested that butyrate and propionate can stabilize T regulatory cell population in the blood. So that is, and then also in that study we noticed that there was a decreased abundance in the memory T cells. So that's where we are heading to with this thought that probiotics might play a role in helping the regulatory arm, which would further mitigate or would counteract this ongoing inflammation in these type 1D patients, as well as their autoantibody negative CYPs.
Very nice. I have two questions for you. Sort of, if we postulate T1D is a relapsing remitting disease in its, progress through stage 0 to stage 3 in diagnosis, how might you establish windows of opportunity to reduce the mono4 NK-like cells?
So that's a very good question. And well, actually, that's the reason why we had this longitudinal analysis done to begin with, because during that longitudinal analysis, what we are trying to do is to study these mono4 or NK-like along with the immunosuppressive T regulatory cells prior to clinical onset. Well, it would have been more interesting that if we could have added more time points, because that would give us a better idea about the changing landscape with having more time points. Then we just have 2 time points. So
more data is always
more
good.
data, yes.
So I guess this follows that question is like, how might you monitor this increase in mono4s and its associated cytokines in context of other infections that these patients may encounter on their way to diagnosis, such as CVB, et cetera. Again, but more time points, right?
Yep.
Yep. So that's really interesting. You have a great model, and it'd be really interesting to see where you guys end up with this. We will certainly be watching, and great work from the Hessner Lab with Tarun Pant today from Medical College of Wisconsin. Thank you so much for sharing, and we will look forward to seeing what comes from the lab next.
Well, thanks a lot, Dr. Wesley, for having me.
Thank you, Dr. Pant. Bye-bye.
You have a good day. Thank you.
You too.